So, good morning, everyone. My name is Sara CARRACEDO. I'm a third-year PhD student. Before to start, I would like to thank the ALS Lab for giving me the opportunity to talk today, where I will present my results on the cell-specific role of p2S4 in ALS and its potential use as a biomarker in humans. What I want to clarify about ALS is the neuroimmune crosstalk that occurs on it, with a first neuroprotective stage where these immune cells protect motor neurons from death, and is following by a neurotoxic stage of these cells which accompany neuroinflammation, the generation of motor neurons, which will release high ATP, extracellular ATP, that will activate many pathways and receptors such as p2S4. So what is the receptor? So p2S4 is an ionotropic channel that displays high calcium permeability, and the curiosity about this receptor is that it's not just expressed in the brain, but it's also expressed in many regions and in many cell types. So when you are expressing many cell types, you direct many kind of functions. So p2S4 has been related to immune responses via microglia and to synaptic plasticity via neurons. But in our team, what we interested the most is about the trafficking of these receptors, as it changes between physiology to pathology. Therefore, in physiological conditions, p2S4 is internalized and kept in lysosomes. Thus, these functions that we observe here are debated in the basal context. However, in pathology, this changes. Now, p2S4 will move to the surface of these cells and will be active, and it will become overexpressed. And this has been seen in diseases such as multiple sclerosis and neuropathic pain. But what happened in ALS? So our team has already started studying p2S4 in ALS, and for that we use the SOD-G93A model, and we study different p2S4 expression chains between cells, and we found that at presymptomatic stages there were upregulation at the surface in peripheral macrophages. And at the level of the spinal cord, there were a switch on the p2S4 expression between motor neurons at symptomatic stages to microglia and other stages. So this change on p2S4 expression that seems to be time-dependent led us wondering whether the modulation of p2S4 could have an impact on ANS. And to do so, we generate new models where we breed these SOD1 mice, either with a model where we increase the surface expression of p2S4, either suppressing the expression of the receptor. And on these models, we found exactly the same. Paradoxically, either increasing p2S4, either blocking, has a beneficial motor performance, and animals were having higher survival. So these paradoxical results and the time-dependent cell movement of p2S4 build what is the main question of my PhD project. From one side, we were wondering if we were able to detect as well in humans this surface increase that we observe in macrophages at presymptomatic stages, and therefore, could we use p2S4 to diagnose ALS? And the second question was, does p2S4 has a more cell-specific role rather than a global function in ALS? So we start for asking this second question, and for doing so, we need new models. So now we will just target directly the surface increase of p2S4 in neurons, or we will suppress its expression, and we will do the same in microglia macrophages. What we did with these animals is different motor tests, such as rotor rod and hanging wire, but today I will just talk about the swimming test and the survival of the animals. And finally, with these animals, we take different time points where we will study the neuronal death and the microglial reactivity. So let's start. What is the neuronal role of p2S4 in ALS? So this is coming a little late. So we can imagine what happened at presymptomatic stages. The animals are able to reach this platform in the swimming test quite fast. However, as the disease is progressing, animals need more and more time until at last stage where the animals are not able to anymore to perform the test. And we observe exactly this on the SOD1 animals. The animals are increasing this time to reach the platform. But what happens when we increase p2S4 in neurons? We observe now that this model needs even more time to reach the platform. However, when we block p2S4 expression, we see again that there is a decrease on the time to reach this platform, and most importantly, the animals are able to seem for longer to perform these tests at later stages. So also, we found a change on the survival of the animals in which the absence of p2S4 in neurons seems to improve the survival comparing with the SOD1 mice, while the KI seems to be more detrimental. So now that we have seen that there is an impact at the behavioral level, what is happening in the spinal cord? So to respond to this question, to answer it, we took the spinal cord samples from presymptomatic, symptomatic, and end-stage animals, and we can observe how we are losing the motor neurons, sustaining growth with anti-CAT over disease progression in all the genotypes, while we are increasing the reactivity of the microglia stained with EVA1 over disease. So we quantify this, and we observe that the mice where we lack pituitary neurons were able to keep more number of motor neurons and with a higher size over disease progression, while opposite was seen to the KI, and we also observed that there is a decrease on this reactivity of the KO animals in the microglia. So now that we know that pituitary in neurons is to be detrimental, what is the role of pituitary in ALS? So we did exactly the same, but now with the specific models, either increasing pituitary surface in microglia macrophages, either blocking its expression. And we found exactly the opposite. Now, this microglia pituitary increase seems to have a beneficial effect in motor performance, while the surface absence, the absence of pituitary in this cell seems to be detrimental. However, we did not observe change at the survival level. And for this monostemisal part, is still a work in progress process. So the conclusions of this first part is that pituitary has dual beneficial and detrimental functions, depending on the neuron that is detrimental and microglia that is beneficial. And the last part of this project was to go now to clinics and to see if we are able to detect aberrant surface changes of pituitary and in ALS patients, and therefore if we are able to diagnose ALS. So we developed a new protocol where we took samples, collect different samples from ALS patients and other patients blind, and we performed centrifugations in this blood to just get this mononuclear fraction, where are the monocytes, which are the precursors of the macrophages in the blood. And with these samples, we did perform flow cytometry analysis, where we stained with two antibodies, one staining the surface of pituitary and the other one the total pool. So we did a ratio. So we have an increase on the ratio, we have an increase on the surface trafficking. And we obtain even this really variability on the samples that this ALS patient seems to increase the surface of pituitary in blood. So these are the conclusions for today. From one side, generalize the role of a protein can occlude some specific effects mediated by specific cells. And in the case of pituitary, we see that this receptor can be both beneficial in the case of microglia and detrimental in the case of neurons. And now we are collecting the samples to perform transcriptomic analysis and see the possible activation pathways that could be involved with these cell changes on the function and how it impairs the motor neuron vulnerability. And on the other case, in order to confirm if pituitary could be a potential biomarker for ALS, we still need healthy volunteers, as all of these were patients, and we also need different forms, genetic and sporadic, of ALS to can generalize this mechanism. And that's all. I would like to thank Wendell and Mason for all the patients' access, and Marie Fakitsa for the immunohistochemistry analysis, and Eric, that is here judging this presentation. So thank you.
Thank you, Sara. Any questions?
Thank you very much. I had a question regarding or whether you are considering any therapeutic avenue using these purinergic receptors. If I remember well, there was a publication from the team of Ludo van den Bosch several years ago that, based on this specific mechanism, that was not so encouraging. So I was wondering whether you had new ideas or whether you only consider it as a biomarker at the moment.
Yeah, about that publication, they have used ivermectin to treat the animals. And ivermectin, yes, is an agonist of pituitary, but also is an agonist of many other receptors. So it was not super specific of pituitary. And with that, I would say that I will not use personally a purinergic receptor, as this is involved in that many cells and in that many regions that maybe you will have other effects that you don't want to in the treatment side. So for the moment, I just consider it as a biomarker.
Thank you.